We have 40 of you joining us today. Thank you so much. We're going to get started in just another minute. We're going to let people get populated, and I'm going to start the talking so we make sure everybody who wants to be here and hear the important news from our friends at Tenaya Therapeutics will hear it, and we'll have a nice population for that. So thank you for joining us today. This is our first special edition Bighearted Warrior 2025. And today we are going to be talking about the exciting topic of gene therapy and a clinical trial update. So here at the HCMA, it's very important for us to let you know when a clinical trial is opening and to let you know how you can get involved. But even more important are these touch points during the discovery process where we get a little bit of feedback to see how things are going. So you're going to learn about that today. As we get everybody populated here, because we have about 170 people said that they wanted to join us. So I want to let those numbers grow a little bit. I'm going to give you some updates here from the HCMA, including that everything after my talk today is being brought to you for informational purposes only. It is not intended to treat or diagnose any disease. If you're interested in learning more about clinical trials discussed in this session, do not hesitate to contact a study coordinator or your physician or here at the HCMA office. Contact information can be found at clinicaltrials.gov at any time or directly on the website of the sponsor or here at the HCMA. We are going to be sending a follow-up survey at the conclusion of this webinar. You'll probably get it tomorrow, the next day. That'll give you an opportunity to let us know if you'd like to learn more about this particular trial. The HCMA is providing this informational sessions to ensure that patients, caregivers, healthcare practitioners, and other interested public members are informed on the inclusion and exclusion criteria of a study and what we have learned from them. We're not going to get too much into inclusion and exclusion criteria today, but we will touch on those topics. We have some familiar faces for anybody who's followed some of our webinars in the past or our podcast. We have Milind Desai joining us from Cleveland Clinic and Whit Tingley from Tenaya Therapeutics. And I'm going to moderate their conversation when we get to the end. And we're going to be hearing about the recently received results from the MyPeak-1 clinical trial. First, some updates from the HCMA so we can get this room nice and populated. Heart Month starts in just a matter of hours, and you're going to be seeing content all month long on our social media feeds, as well as February 26th being HCM Awareness Day. We are going to be having a live event at night. There's going to be games. There's going to be a live event in person, and then there's going to be a virtual event online. Stay tuned for more updates on this. It's going to be a little bit of fun, a little bit of education, and I am really looking forward to this one. We're putting all the pieces together for you right now. If you want to show your support for HCM Awareness Day, we can give you this lovely frame for your social media. All you need to do is send a photo to Olivia and she'll send it back to you in a frame and you can use it on any or all of your social media feeds. Thank you to all of the states that have come on with HCM Awareness Day resolutions, or some states like Ohio has given us an HCM Awareness Day as a law. So sometimes we get proclamations and resolutions, sometimes we get more. So thank you to all of the states who have recognized HCM Awareness Day. We still have time to add more to the list. If your state does not appear on the 2025 list, Julie Russo from the HCMA will be more than happy to tell you what you need to do to get it to happen. Julie's here with us tonight and going to be going backstage in a few minutes. So we can still do this. And in interesting times, we are going to be making our second trip to Capitol Hill. We will be there on April 9th, 2025. And if you are interested in joining us for our legislative briefing on Capitol Hill, you'll need to contact Julie and set up a volunteer intake. Make sure you've done your intake and navigation calls and that you are aware of what we're going for and do your training. We're more than happy to have you join us in Washington, D.C., to ensure that our voices are heard and that the matters that are important to you and your families are communicated effectively to those who write the policies that dictate our healthcare access. So these are some photos from last year. We had a lot of fun. We did a lot of great work, and we're looking forward to going back and making more positive change. If you're interested in joining our ambassador program, this is a social media ambassador or in person, you are welcome to, again, contact Julie or use this QR code and learn more about our programming. It is a fun way to share your story and inspire others. So please join us in our ability to raise awareness. Here's some stuff coming up for 2025. We will be in Orlando on April 25th, Seattle, July 26th, Baltimore, September 13th. Oops, I have a conflict there. Got to fix that one. And then in Pittsburgh on November 15th. So we will be visiting our Center of Excellence partners in those areas and conducting meetings. Seattle is going to be a joint meeting between our two newest members of our HCMA member of family of Centers of Excellence, and that is Virginia Mason and UW. Our annual patient meeting is October 17th through 19th in New Jersey. We encourage you to come on out, hear from thought leaders in the field, start working together as patients to identify the gaps in research, and probably get another update on this particular trial. So the registration will be live soon, but you can save the date now. And I want to thank all of our team. We have a new addition here at the bottom of the list. We have Erica Freeman, who has joined us last week as our communications manager. So welcome aboard, Erica. We're looking forward to working with you. And on that, I always have to thank my donor, Brandy, who left me the opportunity to live a life without my MYBPC3 mutated heart. And today we're going to learn maybe there's some ways that you can deal with your MYBPC3 heart and make sure that you don't have to get a transplant, hopefully. Okay. So I'm sorry, I'm having a technical issue with my screen. Okay, there it goes. And I'm going to stop my screen share and I'm going to welcome Milind and Whit to the webinar. Hello, gentlemen. And Julie, thank you for your time. If you have any technical problems, you can talk to Julie in the chat. And if you have any questions for our speakers, you can use the Q&A box in the bottom of the screen.
Hi, guys. Thanks, Lisa. Thank you for having us. I'm Milind Desai from the Cleveland Clinic. And Whit, you want to introduce yourself?
Absolutely. Hi, everyone. Thank you for joining and your interest in the progress of our trial. Thank you, Lisa, for hosting us, and Dr. Desai for joining us here. I'm Whit Tingley. I'm the Chief Medical Officer at Tenaya Therapeutics and a cardiologist.
Yes. Yeah, I think, and thank you to all the patients and all colleagues, former colleagues, everybody who has joined the webinar. And our hope is to have a fun, informational, educational session about something that is very exciting and which is truly groundbreaking, honestly, in this context. So we are going to divide this presentation into two parts. The first part, Dr. Tingley will walk you through the concepts, how we have gotten here, and then I'll briefly talk about some of the trial stuff. And then we'll have a lively discussion. So Whit, you want to take it away?
Absolutely. I'll do the AAV-based gene therapy 101 part, and Dr. Desai will talk about the progress and initial data that we can share about the MyPeak-1 clinical trial of the first gene therapy for hypertrophic cardiomyopathy. Forward-looking statement is kind of the legal version of Yogi Berra saying that making prediction is hard, especially if it involves the future. So gene therapy is designed to insert a working gene into heart muscle cells to restore the function of that gene and halt the disease caused when those genes aren't working. So these target the underlying genetic cause of familial diseases. The goal is to halt disease progression, reverse symptoms, and improve quality of life. The type of gene therapy we're talking about here, using an adeno-associated virus, or AAV, is administered just once, and then the effects last, we anticipate, for many years. So a single IV infusion with the potential for a long-lasting response. We get a question a lot about whether, if you're treated, you pass on the treatment to your children, and the answer is no. The gene we deliver is not passed on, not inherited by your children. There have been many successes in different therapeutic areas, different types of diseases with gene therapies, and over 5,000 people treated today with different forms of gene therapies. For the sake of time, I'm not going to go through each of these, but I will say that we are making steady advancements in gene therapy for heart disease and cardiomyopathy, with encouraging news coming for patients with hypertrophic cardiomyopathy due to Danon disease and Friedreich's ataxia as well. So a very exciting time for development, but for patients as well. All right, today we'll talk about a specific type of hypertrophic cardiomyopathy caused by mutations in the gene MYBPC3. This affects about 120,000 people in the United States alone. It's the most common genetic cause of hypertrophic cardiomyopathy, and the cause, of course, of Lisa's hypertrophic cardiomyopathy. So mutations of the gene prevent a key protein called MYBPC, or C protein for short, from being manufactured by the cell. This protein has a critical function regulating the contraction of the cell. So the proteins that cause contraction are easily thought of as hands pulling a rope. On the illustration here, you see the blue myosin head, that's the hand, and the rope is the light brown actin. The disease happens when there are too many hands pulling the rope. And the C protein, or MyBPC protein's job, is to regulate the number of hands pulling the rope. And genetic mutations cause there to be not enough of the C protein, too many hands pulling the rope. So our goal is to increase protein levels, then restore normal regulation of the normal number of hands on the rope, to prevent the thickening of the left ventricle, to improve relaxation of the ventricle, and to prevent ventricular arrhythmias as well. So I'll start with a high level how this works, and then I'll go into an analogy about how gene therapy can deliver genes to the heart. So we are, with our gene therapy called TN-201, targeting the underlying genetic cause of the disease. And we use what's called a capsid, like a box that delivers the gene. So this capsid is made of proteins that viruses have used to get their genes to the heart cells, but all the viral genes are not included. They've been eliminated. And instead, we put in the functional gene, in this case, the MYBPC3 gene. When this gets to the heart cells, the gene goes to the nucleus and it's turned on, and it's expressed as mRNA and protein. We'll get back to those. This has the potential to halt disease progression, reverse symptoms, and improve quality of life. And we'll talk today about the first in human studies to begin to get information about whether it does those things. All right, here's the analogy. It's basically a do-it-yourself furniture building project. So I'll talk about the furniture first and then go through how this is how the analogy works. But imagine you don't have enough chairs for your tables, and you're hosting a party, and you're going to build the chairs yourself. So you order an instruction manual. So the company puts that manual in a box and delivers that box to your house. You bring it inside, you open it up. You make copies of the instructions for your friends so they can all help you making the chairs. You put them together and you put them around the table. So that's the analogy. So in this analogy, the chairs are the C protein, the MYBPC protein, and your heart doesn't have enough of that protein. The instructions are the DNA, and the box is the viral proteins, the capsid that I talked about before. So we at Tenaya manufacture the gene therapy. We're putting the instructions in the box, and then that is delivered to the patient as an IV infusion. And so the IV infusion is given. The box circulates around the body and then goes to the heart muscle cells. So that's the house. And then the cell takes it up inside. It's brought to the nucleus. And then the instructions end up inside the nucleus of the cell. Then the nucleus turns the DNA instructions into messenger RNA. This is a way of making more copies and essentially translating the instructions into a way the cells can use more practically. So DNA is the gene, the full instructions. The mRNA are the copies of the instructions as it's going into effect. And then the cells use the messenger RNA to make the protein, the C protein that we're trying to make here. And they use parts that are in cells already called amino acids to synthesize the protein or make the chairs. So that's fundamentally how it works. Happy to answer questions about that at the end of the session. There are, of course, potential risks to gene therapy. One important one is the immune system recognizing the box, the capsid. It can think of the box as a foreign invader and tries to reject it, and that can make it so that the treatment doesn't work as intended. And in cases, this can cause liver inflammation that is manageable, and we'll talk about that more in Dr. Desai's section. Currently, a patient can only receive AAV gene therapy once. Because of the immune system recognizing it, you develop antibodies that would prevent future doses from working. So patients can't be redosed with higher or the same doses in the future. These boxes aren't perfectly targeted to the heart. They do deliver copies of the gene to other tissues and organs. We use a technique called a promoter to drive the expression of the C protein just in the heart cells. And of course, these gene therapies are still under investigation, and not all of the potential risks are known or have been identified yet. So with that, we'll get into the initial findings of the MyPeak-1 clinical trial. Dr. Desai.
Thanks, Whit. Man, this is kind of like IKEA here. You deliver the box to my house, and then we have a party trying to figure it out. That's a novel concept. I mean, I'm gonna use that. I like how you presented that. It makes it very easy to understand. But thank you, Whit, and look, thanks again for everybody who have joined. More importantly, I'm gonna start by thanking the brave patients who have volunteered to be part of this, okay? Cannot underscore the importance of that. The first patient, when they were dosed, I was looking straight into their eye and say, this is like going to the moon. That's it. I mean, there's no other analogy that I can think of. This is like going to the moon. We have scienced our way. We have scienced our way to this point, but we still are going to the moon. And, I don't know about the patient, but I sure as heck was having palpitations. And I slept literally no more than 2 hours the night before. I was up pretty much, am I doing the right thing? But looking back at it, yes. So what are the highlights? We are, for the record, Lisa, we are gonna discuss the data in greater detail at American College of Cardiology, where this work has been accepted as a late-breaking featured science presentation. Okay, so more data forthcoming. But what are some of the big picture highlights? Let's start with safety. This is a phase 1 trial. So the most important part of phase 1 is what? We need to appreciate the dose, and are we doing things safely? So safety is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 for me. It was well tolerated, and safety profile was consistent with other gene therapy. Now, for the patients in this webinar, after every patient dosing, or every day after the patient was dosed for the first week or so, we would get together, and after every dosing, there was intense discussion, and a DMC, Independent Data Safety Monitoring Board of expert panelists, endorsed proceeding to the second planned dose of TN-201. The other important thing is, the piece of furniture was sent in the mail from IKEA. We needed to make sure it was delivered to the heart. So biopsy of the heart, endomyocardial biopsy, was very crucial. And we have shown that the infusion does reach the heart cells, and it is expressed. Okay, it is delivering the gene, the DNA. the working copy, the DNA is converting into RNA, and RNA is helping generate protein. Obviously, these are early days. We are trying to figure out what is the optimal dose, what is the optimal protein level needed. So this is an ongoing study, and we are just presenting very early results. And clinical endpoints are encouraging, but as always, these are early and further follow-up is needed. Next slide. So what are these folks, the three brave individuals? All these are my patients, okay? Ironically, not one of them is from Ohio. Not one of them is from Ohio. And compared to typical HCM patient, these were younger and more severely affected, 2 females and 1 male. Mean age, as you can see, the average typical age of an HCM patient population is 50 years. These were much younger. Based on the early study protocol, everybody had an ICD. These protocol requirements are evolving. Importantly, all 3 patients had a myectomy at an experience center outside of the state of Ohio. They had a successful myectomy to relieve outflow tract obstruction, but they were symptomatic. They had very high to high levels of residual hypertrophy, and they had limitations, residual heart failure symptoms. So the message here is myectomy works to relieve outflow tract obstruction, but there may be problems beyond myectomy that at least in these 3 individuals we had. Next slide, please. So, the other way. Yes, so no, go back, sorry. Yeah, so initial safety profile, what did we find? This is an infusion, and we expect liver enzymes to go up. So they went appropriately increased. They were appropriately increased. They were temporary, and there were no surprises. These were very typical of what you would expect from gene therapy infusions. The first 2 patients have completed steroid treatment. So steroid treatment is necessary as part of these gene therapy. It's kind of like Lisa, who's a transplant survivor, knows immunosuppression is par for the course. Patient 3 is tapering off as we speak. A big worry is something called thrombotic microangiopathy, where you develop spontaneous blood clots in your blood microcirculation or loss of platelets. That's a major problem that we are looking for. Expected known complication of gene therapy, but no patient had this. There were no signs of heart toxicity in the 3 patients, myocarditis, no abnormal heart rhythms, no ICD discharges, stable ejection fraction in all these patients. And none of the patient has discontinued the study protocol. We plan on following these folks for a total of 5 years. They are all doing well. Majority of the health events, adverse effects, were mild and short in duration or reversible. Based on the protocol criteria decided, there were 2 what were deemed as serious health events, but that were deemed by the DSMB to be by the Data Safety Monitoring Board to be unrelated to TN-201 gene therapy. Next slide, please. So the important things, were the goods delivered? Biopsies from the first 2 patients show that the gene was delivered successfully to the heart. Okay, high level of gene delivery in both the patients, and the delivery of the gene was above the amount needed in animal studies to restore human functions. Okay, so that is good news. At least the goods are being delivered where we think they need to be delivered. Next slide. The next step, as Dr. Tingley had mentioned, the DNA, the box, the IKEA box is there. Now you have to unpackage the box and start to assemble the furniture. That step is the RNA expression, and RNA has been actively expressed in both the patients. Expression is similar to other gene therapies for the heart. It is lower than in mice at the same dose, but expected based on species to species therapy of variability. And you can see in the bar graphs, it is in the range of where one would expect. More time, obviously, and more study participation is needed. How much RNA expression is needed to restore, to generate the protein enough to restore the function for a long time in patients with this condition? Next slide. So the other important aspect, because of the positive signal in the first 3 patients, or the cohort 1, the DSMB met and approved us to proceed to cohort 2. And we are pleased to inform that the first patient has been dosed at the higher dose in cohort 2, and the enrollment is ongoing. Additional data, as I alluded to, will be shared, including additional biopsy results, et cetera, in the first half of 2025. We can now say, certainly starting at ACC and American College of Cardiology, and cohort 2, hopefully we anticipate in the second half of 2025. Next slide. So to summarize, as endorsed by the Independent Data Safety Monitoring Board, which is world experts in the field of immunotherapy, immunosuppression and gene therapy, TN-201 was well tolerated and safety is consistent with other gene therapies. Biopsy proves that the goods are delivered. The working MYBPC3 gene is delivered to the heart, and it is expressing mRNA. Clinical endpoints, we are still in early days. We do not want to put the cart before the horse, but certainly stability is observed, and to an extent, improvement in certain parameters is observed. We clearly need to further continue to follow these patients and enroll more in the group. Next slide. This is the infomercial piece. You can go to all the sites. Is TikTok there? It may or may not be working, but it is what it is. So look, as patients, this is a partnership. This is a partnership between a committed sponsor, the patients, motivated patients, and academic institutions and advocacy groups for us to move the science and the field forward. And I sincerely appreciate all the things that are happening and look forward to what we can do more together. I thank you very much again for joining, and I think it's time to have a rock and roll conversation.
So I am going to ask Whit to take the slides back to the IKEA furniture for a minute. I want to do that again, because I think it was a really good illustration. And if people didn't quite catch it the first time, I thought maybe we can kind of go through the steps again to explain this process, because I think this is an elegant way to explain a very heady concept with what we all know. Like, we've all put together the IKEA furniture or the whatever, and we know how complicated yet simple that process is. And I think this illustrated it very well. While we're going through this again, I'm going to tell you that this is the time to post your questions in the Q&A box. I am absolutely dumbfounded that there's no questions in here yet. So I just need to make sure y'all know where to find those. Oh, there we go. Benjamin, you didn't let me down, bud. Okay, here they come. So you populate those questions, and we're going to go through the IKEA catalog again. Dr. Tingley, the stage is yours.
Yeah, I'll just try to frame it a different way. So that really the only thing that is missing for you to make your chairs are the instructions. So patients are born missing instructions. The gene is mutated, it's misspelled, the instructions don't make sense. All the parts are actually there. When you eat protein, you're digesting amino acids. Those are the parts that are needed to make the protein. But the instructions are missing. So we just need to deliver the instructions to the cell. So we deliver it just like an IKEA delivery. We're going to put the manual in the box, and the box has the address to your house. So the box is made out of protein. It's called the capsid. In this case, it's called an AAV9 capsid. And it's got a label on it that says go to the heart, just like the delivery address. Again, it does go to other tissues as well, but a major address on the box is the house, the heart muscle cell. And then in order to make the furniture, of course, you got to bring the instructions into your workshop or wherever. That's the nucleus. And open the manual. And in this case, the cell needs to simplify the manual and make extra copies. So that's what the mRNA is. It's like another way to think about it is this manual is like your favorite recipe book, and you're going to Xerox the copy of that recipe so that, when you take it into the kitchen and work with it, if it gets stained, your book is fine. So that's sort of the mRNA process. It's like the copies made off the master instructions. And then the cell puts together all the parts, the amino acids based on the instructions in the right order to make the MYBPC3 protein or the chair here. That's that's
I think that's excellent. And in that time where you explained it again, and I think it makes a lot of sense, the Q&A box is filled up. Now I am going to start moderating the Q&A with a bit of a statement / disclosure. There may be facts known to the company and to an investigator that they are not permitted to disclose at this time. So if you ask a question and they say, we can't respond to that right now, they're not being difficult. There are regulatory processes here that we must adhere to. And when they release data, that data is released in a very specific format for a lot of reasons, including SEC regulations. So if we say we can't answer you right now, please don't read anything into it. The data will all eventually come out. And when they hear your questions, I'm sure that'll encourage them to maybe get that data to us a little faster. That I am not speaking on behalf of Tenaya for that one. I'm just being snarky me. Benjamin, who started us off, how do you determine what the ideal dose is to achieve the desired level of heart function? And how do you measure that it's actually reached the muscle?
I'm happy to start with that one. So we have learned from the Danon gene therapy studies and others that the dose in patients is very similar to what's needed in the mouse model. How could that work? Because mice are so much smaller. Well, these are weight-based doses. So when you figure out the body weight and then you calculate what the dose is for the body weight, that number actually ends up being similar from mouse to patients. So when we designed our study, we chose a starting dose that we expected and hoped would be an effective dose for those patients. So we're very happy to see we are delivering more than enough DNA than we needed in the mice.
So yeah, and again, I don't have much more beyond what Dr. Tingley said, but this is where the concept of the phase 1 study is. We have the lower dose cohort, then we are gonna test the higher dose cohort, and then once we have looked at their results, including biopsy, etc., we will get a sense of where we fall, where we should fall, and we always have to balance that with safety. So, more is not always good. There will be an optimal threshold which balances the good and the not so good. So more to come, I guess.
More to come. Okay.
So that's the second dose cohort. We'll be able to compare the doses, compare the level of DNA, RNA, and ultimately protein, and choose which is the best dose to take forward into future clinical trials, including the ones that would support approval by the FDA.
You kind of read my mind on that. I'm like, what does this all mean? Like, how do we ramp up? So I think that was a great explanation. Thank you. I don't know that this one's going to be answerable, Deborah, so I will ask, how are the patients from cohort 1 feeling? Are they noticing any changes in daily symptoms?
So I will take that answer. So this is how I'm going to answer it. After every patient dosed, there was a committee of experts who told me, okay, whether to continue to the next patient or not. And we were told to continue to the next patient. So read between the lines that, if the committee felt that there were any major safety or efficacy concerns, they would have stopped us, and they did not. We can move on to the next question.
I will say to that question and what Dr. Desai just said here, there are signals that I look for as I am trying to evaluate where we are with things. And the minute a DSMB comes back and says, you can move forward, and oh, by the way, they don't need to have an ICD. They don't have to be post-myectomy. They don't have to be obstructed. These are signals that are telling me things are going in the right direction. This is the extent that I will say it at this point. And I think that's an indication of such. Heather said, that helps, but I'm not quite sure what that meant, but glad that it helped. So Dr. Desai, I'm going to ask you to clarify the second part of this statement / question from somebody. Is this only for people who have, quote, familial HCM? I was told I do capital, not have the inherited kind. So can we talk about the importance of knowing one's genetic marker for HCM and break it from there?
Yes. So, yeah, as Dr. Tingley presented, this specific gene therapy, this specific construct is for MYBPC3 mutation, even there where there's the mutation which causes loss of function, meaning MYBPC3 loss of function, which reduces the amount of protein that is delivered. Okay, so this one we are still in very early days of gene therapy, right? So this is specifically for that mutation. At this point of time, this therapy is not for a person who does not have this mutation or who does not have an identifiable mutation. That is not to say that the world is not moving towards identifying newer constructs, newer mutations, and newer therapies. So, this is step one. Okay, this is step one. Think of this as inventing Model T, or rather inventing the first car in Germany, right? We are from there. We are now talking about potentially autonomous cars and maybe flying taxis. So we are just getting started. Okay, and this is the future is bright.
Yeah. So for this gene therapy to work, your hypertrophic cardiomyopathy has to be caused by a mutation in the MYBPC3 gene. It doesn't have to be the case that you have a family member diagnosed with the disease. It might be that, unfortunately, early on in your development, you developed the mutation and you're the first in your family, or it could be that a member of your family had the mutation and just hasn't been or wasn't diagnosed. So you don't need to have familial in the sense of like demonstrated multiple cases in your family, but you do have to have a pathogenic or likely pathogenic mutation of the MYBPC3 gene.
Yeah, perfect.
But not a VUS.
Not a variant of uncertain significance.
Yeah, not VUS. No.
Okay.
Absolutely.
So, Whit, thank you. You said everything that I was going to say. If you didn't say it, 100% accurate. And please listen very carefully to one last statement here. If you have not had genetic testing for HCM yet, you need to speak to your team about getting your genetic testing. Not only can you find out if you might be eligible for a clinical trial, but then you can also use that information to see who else in the family might be affected. You can also use that information in your choice to have a family, and you can use something called pre-implantation genetic diagnostics. There are a lot of things you can do. You can also find out that your HCM is not really HCM. It's another one of the mimickers or phenocopies, amyloidosis, Fabry's disease, RASopathies, Danon disease. So you can find out what path you should be on in terms of your treatment journey. So it's really, gene testing today is really about the diagnostic journey. You don't really know your underlying cause until you go through all the steps. 60% can't find a gene yet, but someday we're going to figure out what that is, and it's probably going to answer a whole bunch at one time. So if you're an unknown gene, I know it seems a little frustrating that you didn't get an answer, but you did get an answer. You don't have an identifiable gene. You may later qualify for a trial or an investigation into what those other genes might be. So it's still important. Next question. Can the doctors please discuss some of the side effects experienced by the patients who were dosed?
So the most common, I mean, the most common is us being hypervigilant of their blood levels. Okay, the most common thing we are concerned about are these liver function abnormalities, and we are watching those like a hawk. We are also, there's something called complement levels. These are fancy blood tests that that help predict if you are going into the dreaded complication of thrombotic microangiopathy, etc. So those, I mean, and we did not have those. The LF liver function test was the most common situation. Now, for the first 3 patients, we basically observed them in hospital for a few days, and it sort of, they did not feel too different, for the lack of a better term, during their initial hospitalization. Of course, these folks are on immunosuppression. Okay, so think about it. So you are at a significantly increased risk of infection and etc. Now, I will actually, I take it back. One of the biggest side effects that I heard consistently is these folks are on high dose of steroids, so they felt buzzed. They felt buzzed all the time, even at night.
And hungry.
And hungry and nighttime. And I could walk in and I'm like, wow, your face is now nice and chubby kind of thing, the steroid face. So these were some of the most common things that we observed. But make no mistake about it. Secondary infection. Think of yourself as somebody who just had a transplant. Lisa, you've gone through, I mean,
Yep.
you are at a significantly increased risk for secondary infections. So I would say that's generally how I would look at it.
Yeah, and I'll
Okay.
say that a difference between transplant is that we can stop the immunosuppression.
Yeah.
So as I said, one of the potential side effects of gene therapy is the immune system reacting to the gene therapy. It's reacting to the box, and
Yeah.
the box gets destroyed, just like you throw out or recycle your IKEA box. So there's only a temporary period over weeks to several months that box is around, and that we need to use the immunosuppression. We do need steroids at the beginning, but we start fairly early, decreasing the dose and then watching the liver very closely as we work quickly to get off the steroids.
Yeah, that's an important point. Yes.
Okay, we've got about a dozen more questions to get through, but some of them connect, and we've already answered Bill's to a certain extent. Are any people in the cohort older or without surgery? So you saw the slide of who was in this trial, but the DSMB has now opened it up to, so here we have the average, and then a 27-year-old female, 43-year-old female, 47-year-old male. Ladies are in the lead, just saying, ladies are in the lead. That's who we had here. I don't know the demographic makeup, nor do I know that they can share with us of this higher dose cohort person being entered into the trial. But we now know that you don't have to be obstructed, you don't have to have an ICD. And the age limit for the trial, if somebody could remind me,
Yeah,
is
this is for adults.
Yeah.
It was initially just for people with non-obstructive disease, but that included people that had obstructive disease before and had a surgery that removed the obstruction, which is the case for all 3 patients. So the surgery wasn't required, but we have opened up the criteria, so either non-obstructive or obstructive people qualify for the trial now, all adults. Initially, we required an implantable defibrillator, or ICD. There's no connection between the C protein and arrhythmias, no causal mechanistic pathway there. There wasn't anything seen in animal studies, and nothing reporting out of these ICDs, and no events from the patient. So in the FDA, we removed that requirement from the protocol.
I mean, look, the way I look at it is you are embarking upon a first in human adventure. You better err on the side of caution. And that's what we did, but we have learned enough that we feel confident that not having an ICD is okay. And expanding it into broader indication is okay.
it's okay. So the question is, are there trials for other genes in the work? And the answer is yes, very, very, very many. Within the space of HCM, right now, there is this trial. There is the Dannon's disease trial by Rocket Pharmaceutical, which we're learning a lot from. The specific question is, are you aware of anything in Fabry's disease with gene therapy?
Yes, that's working for Fabry's as well.
There's one in for Fabry's. So there are we've been talking to a lot of companies interested in a lot of things, all genetic. I know some people are planning things for MYH7. Some are planning some for the troponin mutations. There's a PRK mutation that's looking at a therapy, a BAG gene, which more is a dilated cardiomyopathy. And I'm trying to think of them all off the top of my head, but there are many. Stay in touch. We will probably be building out a page on our website with all the active genetic trials going on. And we are considering at the HCMA submitting a letter of intent to the FDA to run another patient-focused drug development meeting specifically about genetic therapies in the big-hearted space. So stay tuned on that. I'm not quite sure if PFDDs are going to be a thing next month, but we're ready to send the letter in, but we're just kind of holding our breath for another couple of hours to see who the Department of Health will be running by. So we'll just leave that there. So that answered your question. Are potential study participants being proactively contacted to discuss enrollment? If so, how does that process work? If not, why not? So if you have already told your team that you're interested in being in a clinical trial, you've then met with a clinical site coordinator to go through a consenting process to be screened. If you have not done that and you are interested in finding a site, you can call us at the office. You can call Tenaya directly. We're going to send you out a survey, and it's going to ask you for your permission to share some contact information with a site coordinator. So we're working on pulling those pieces together for you. So there's a lot of different ways you can do that. Whit, Milind, did I miss anything?
No, I think, yeah, there's lots of ways, I mean, Lisa, your advocacy group, Tenaya has a patient-facing.
Tenaya has Wendy. Like
Wendy,
that's all they need.
exactly.
They
I
just
was
need
gonna,
Wendy.
you stole the thunder, Wendy Borsari, who's on the call right now, and she's a great resource. You can reach out to the sites if you have a relationship with the site. I mean, there's plenty of ways you can reach Rome.
And I'll say that a great resource for patients interested in trials that might be appropriate for them is the clinicaltrials.gov website. So there you get access to all the trials across all diseases, but there are many for hypertrophic cardiomyopathy that you could potentially participate in.
So because that site is just so easy to navigate as a patient,
Mm.
we have a tool on our website called Antidote, which you can put in your demographic information. You don't have to give them contact information, and it'll identify trial sites near you just for HCM. So you can use that tool as well if it makes it a little easier for you. And I know that there are travel vouchers and travel funding available if you do have to travel a distance to be at one of these sites. So if you have the time and you have the interest
Yes, we
and you
cover
are the
all
right
of
patient and you screen in, there's a way. There's a way. What is the process and timing for expanding the trial to more patients to participate?
Yeah, so as Dr. Desai explained, at the beginning of each dose level, we treat patients one at a time and then review safety and efficacy and make decisions about that. And then at the end of a cohort, we have decisions about dose escalating or continuing at that dose level. So the next decision will be a DSMB meeting to determine whether to go beyond 3 patients at one or both dose levels. And the protocol allows up to 24 more patients when we expand. And when we expand, we no longer dose one at a time. We can dose people much closer together.
I think, and again, to Whit's point, look, this is rightfully cautious treading right now till we ascertain safety and comfort on both sides. And then, then it will, I cannot wait for the day, which I'm hoping is coming soon, where a highly patient person like me, joke there. I mean, doesn't have to wait for the next dose patient to be opened up. I mean, I want this to start quickly and where we can enroll. But we gotta do stepwise. We gotta do the right things first. We gotta ascertain the safety here.
So slow and safe.
Yeah.
So we can gain
Yeah.
confidence,
Absolutely.
understand the potential side effects, evaluate whether it's getting to the destination, and then prove our point, move on faster.
And look, I will say this for the record. Every patient, every experience I have tweaked my thought process, approach, and we have, simple things, as to starting a midline versus a pick line versus, doing the, subtle things that you don't think about till you are walking the walk. It's a learning experience. It's a learning experience.
Okay, we're going to go rapid fire now because I don't want to run out of time and everybody's got great questions. What kind of time is needed to know if the improvement is solidly in place? Part 1 of the question. Part 2 of the question is there federal funding here? Part 3 of the question is, how do you select the location for future cohorts? Well, okay, so let's take this in pieces. How long does it take to determine that the capsid goes to the heart and does a good thing? Simplified.
Yeah, so the DNA data we showed was from 8 weeks after the single dose, which is early for gene therapy. We know from other data that DNA is still there after a year, and we know from the Danon study it's still there after 3 years. And the way the DNA gets in the nucleus and stays there, it could last for many more years after that. We will follow the time course more carefully with more biopsies and more specimens going forward. But given the data, it also lasts a long time in animals as well. So all the data suggests that we can measure the DNA in the first 8 weeks, and then that it lasts for long periods of time. Now, the other part of the kinetics or the time course of activity is the expression, the turning on, the making the messenger RNA or the copying the instruction manual and making the protein or the chairs. That takes longer, probably months. We have encouraging signs over the first year. We look forward to sharing more data about that, but it could take even longer than a year. And we do know that there's now a scientific publication on that first study in Danon disease. And we see many patients where there's more protein accumulating in the second year and more improvements in the heart over the second year. So we do need more time following these patients and more data from more patients enrolled at both those cohorts to give a really full picture of the time course of improvements and the types of improvements that we might see.
Is there any federal funding at Tenaya Therapeutics?
No, no risk from the current freeze.
Situation, yes. And I'm gonna reframe Dolores's third point of her question. How do you select the locations for future cohorts? There are many clinical trial sites that are contracted and ready to do the work. And the site isn't as connected to the cohort as your question makes it sound.
Yeah, I think, you know, I can address to a little bit of that is, you know, obviously, if a site that is capable, that is contracted, that is up and running, that has all the bells and whistles in place, that makes logistically the most sense for you from a geographic distance perspective, I think makes the most sense as a patient. Having said that, you know, I just told you started there, you know, I live and work in Cleveland, Ohio. Not one patient mentioned here is from the state of Ohio, right? So it is, you, the patient, have to be comfortable with the process. And one thing I gotta tell you, you know, it's again kind of like going to the moon. Once the rocket launches off, you can't change your mind and say, hey, can I turn back and get back to Houston? No, you know, once the infusion is in, right? So all these things have to be carefully thought of and discussed, shared decision making.
Completely.
Yeah.
We have
So
two questions.
We've identified expert centers in various parts of the country to minimize travel burden, but we fully reimburse travel. And we very much look forward to expanding outside of the United States with our next trial, the pivotal trial that we talked about before.
Okay. We have two questions on MYH7. So somebody said, is this a potential treatment for MYH7? And somebody asked, how is this therapy applicable to other variants like MYH7? And as it is the second most common mutation in HCM, I think it warrants a little discussion as to what's coming down the pike for MYH7 and why that approach is a little bit different. Whit?
Yeah, so TN201, this gene therapy won't help people with MYH7 mutations. It's a different protein. And in that case, I'm trying to go back to that key analogy. But it's basically the chairs around the table are broken. And you actually need to fix the chair, not build new chairs. And that's a very different technology or type of technology. So there's really important work being done on that by both academic groups as well as industry. So it takes a little bit longer because of this different need. But certainly we're all taking the needs of these patients very seriously.
And you know, in simple terms, you know, this is how I look at it. There's the piece of what exactly the mutation does to result in HCM. We have to account for that. So Whit mentioned MYH7 versus MYBPC3. Then there's the delivery mechanism. Is it going to be the same? Is the capsid big enough, or is the payload going to be heavy enough that we have to re-engineer the capsid? And then the other thing is the immunosuppression piece. So, you know, it is not going to be a one size fits all, obviously, but you know, this science is rapidly evolving where we're gonna science our way through this.
Okay, we are at time, so I'm going to do rapid fire. I'm going to attempt rapid fire with Milind Desai. Ladies and gentlemen, let's see if it's possible. Milind and I can talk for hours together. Okay, so there's a couple of questions. You mentioned sites out of the country. Do you have any Canadian sites that you're considering? If not, I think somebody might be interested.
We will accept patients coming in from Canada. We do not have an active site in Canada now. With our next trial, we will have more sites in a broader global.
So an international patient can come to the United States and get into the trial.
Yeah.
Yes.
Great clarification. Thank you. Are you allowing statins on these patients if they come in?
Yeah.
Yes. Any? You're good. Got your statins. Is there a specific MYBPC variant that is an appropriate candidate? And are there MYBPC3s who aren't appropriate candidates?
That's rare, but that does happen. So part of the screening process is repeating the genotype at our core lab and confirming that it's the appropriate type of mutation, which will be the case in 90% of patients or so.
I gotta tell you, Lisa, it did happen in one of my patients, and I don't know who was more disappointed, me or the patient, because whatever. Yes, it did. So it happens. Yeah, patient had a frame shift mutation, and was not a candidate for it, or at least not at that time. It may evolve to a point where he may become an eligible candidate.
Okay. Okay. Does the hand on the rope analogy work for explaining other kinds of HCM mutations as well?
Yeah, the MYH7 we just talked about is the hand. And those mutations, some of them prevent it from interacting with the MYBPC3 protein. So that it is a pathway for many gene mutations resulting in too many hands on the rope. So that's how different genes can cause hypertrophic cardiomyopathy. Now, there are other very different causes. We talked about Danon disease, Friedreich's ataxia, very different types of hypertrophy, nothing to do with the numbers of hands on the rope. But there are several genes involved called the sarcomeric genes that are involving the hands, the rope, and the regulation of the number of hands.
Fantastic. If somebody has multiple mutations, can you just go after one of the mutations?
So if someone has two pathogenic or likely pathogenic mutations, this will only treat one of them if it's the MYBPC3, and it would be unclear how much someone might benefit from that. So in the current trial, you're only eligible if you only have mutations in MYBPC3. We're not taking patients currently with many genes. But as we learn more and determine the efficacy of this gene therapy TN-201 for patients with MYBPC3 alone in the future, we would try this in more complex genetic backgrounds. But we want to demonstrate safety and efficacy first in the patients that are most likely to respond.
These next two questions are very heady. So let's just be aware of time as we're going to give answers, but they may take a little bit of time. If after receiving the initial dose, the cell makes copies to produce the protein, why increase the dose in the next arm of the study if the copying process becomes self-sustaining? Why do you need to look at a higher dose?
Whit, you can take, but I'll give you my clinician scientist perspective, and that is, we don't look. We are greedy. We want to see what is the maximum of allowable safe dose that will produce the maximum amount of protein at a safe level. Because right now we just don't know. I mean, the dose that we have started off, it is, let's say, it's generating only X amount of protein, and we realistically need 2.5X. But we find out, so we have to sort those things out.
Yeah, and we are delivering robust levels of DNA. In people, the DNA doesn't turn on as effectively as it does in mice, so there is room to produce more mRNA, and we're still learning more about the protein. So we definitely want to compare different doses, and that's just a fundamental principle of drug development, is try several doses and choose which one is best. And we do this in early development and then test the best dose in the pivotal trial for approval.
Yes, and someday I'll have my niece tell you about her experience in a clinical trial in cohort 1 or cohort 3, finding out how high they pushed the dose. That was interesting. So you need to find that that bracket and what's the low and what's the high, and mama bear finds the right bed. So we got to keep trying. It is assumed that once the DNA is delivered, there would be remodeling of the heart muscle, as it does in remodeling things like myocardial disarray. Are we going to remodel the muscle and create more myocardial disarray, or are we going to teach the sarcomere how to act properly and stop causing trouble?
Early days. But the remodeling, the concept of remodeling, we are shooting for the good kind of remodeling, the favorable kind of remodeling, which results in mass regression. So the thick, juicy, hypertrophied cell becomes thinner. We are hoping that once that happens, the heart overall gets less stiffer and functions more efficiently, uses energy more efficiently. The row bars that Whit showed about. So I think that's the expectation from this trial, from this therapy. Whit, you wanna?
That's certainly what we've designed it to do, and the tests in the animal models show that. So even after disease has been established, we see improvements in heart wall thickness and heart function, so the beneficial remodeling that Dr. Desai said. Now, people aren't animals, and we need to do this trial to figure that out, whether it works that way, but that's certainly the hope.
Wonderful. This is a long one, so stick with me because it's a really good question too. Are virus capsids the only vehicle to introduce therapy to the heart? I understand that there is no viral DNA material in the virus capsid. Just curious if there are other vehicles being looked at for gene therapy, or do virus capsids have the ideal shell for gene therapy? We got smart people out here today, guys. We got some smart ones.
So the viral capsids are the best we have today to deliver genes to the heart. They are not ideal. They're too small, so only smaller genes fit. And so other genes like titin is a major cause of dilated cardiomyopathy. That is much too big. Myosin heavy chain is big. There are others. So it's too small and we need new technologies. So lipid nanoparticles is one type of technology being developed, but those don't naturally go to the heart. And so further improvement is needed in that regard. So there's a lot of work in the industry and in academics to improve delivery of genes to the heart. But we're very fortunate to have the AAVs, in particular AAV9, which has now been demonstrated to be very effective at doing this for the right genes.
And again, look, this is the beginning, right? This is the beginning. I think the computers that send the man to the moon was a fraction powerful of
That's
what
what's
our
in your cell
iPhone.
phone. Yeah.
Yeah. So we are just getting started.
Okay. All right. So we're good. No more questions. We are done. Any further questions, you can email support@4hcm.org and we will get your question to our speakers. So we're closed down on questions now. There's only 2 questions and 2 comments, really. You're welcome, Tim. Is there a risk of too much protein production? If so, what could that effect look like? Can we overcorrect?
Right. So we are carefully monitoring, and we will certainly look. And if we see that, the dose that we choose, but we don't expect to see it, and we don't know what harm it could cause, even if we did see it. So we have given animals much higher doses than we're using in clinic, and we don't get excess protein. So heart cells are basically removing the extra protein very quickly. And there's no known function of the MYBPC3 protein except binding the hands and the ropes. And there's just a limited normal physiologic number of binding sites, and there's nowhere for it to go when those binding sites are full, which is when there's a normal level of protein. So it's actually very hard to generate too much protein in the animal models. Human hearts work very similarly, so we do not expect to see too much protein, but we do monitor carefully.
Okay. Can you get a recording of this session. Yes, it's live on Facebook right now, which I'll be talking to our Facebook friends in just a hot minute. So you can see it there, and it will be on our YouTube channel, and it will also be housed on our website shortly. And FYI, HCMA is moving away from YouTube, and we are embracing Vimeo. We are a new user of Vimeo. So we will be having some changes to our website, including our video content. So you'll be able to catch it all on our Vimeo channel. So you don't have to see advertising from somebody else while you're watching our content. So you'll be able to get it Facebook. It'll be on YouTube, but then you'll see it on our Vimeo site as well, coming up in a couple weeks. Thank you for answering that. You are very welcome. Tim, thank you. You're welcome. Thank you, thank you, thank you. So we got lots of thank yous out there, but we're going to talk to the people out on Facebook for a minute here. I'm just seeing what's going on out there. We have a hello, Reinhardt being symptomatic once working MYBPC delivery. Being symptomatic and once the MYBPC gene is delivered, can someone expect symptom reduction and cardiac function changes? So that's kind of what we're shooting for.
That's what we're
Yeah.
shooting for. Yeah.
That's the goal.
Even-
Would apical HCM be appropriate for gene therapy if they have myosin binding protein C known mutation? Yes. And somebody would love a trial in Canada. So that's what happened out on Facebook while we weren't paying attention to the questions on Facebook. So we have some viewers over there as well. Milind, Whit, I thank you so much for your participation this evening and helping explain this very complicated and incredibly important research project and clinical trial to the patients who are going to benefit from it the most, and some of our partners out there in the clinical world and industry as well. They're all watching. We're all very interested to see where this goes. As an MYBPC3 family that's growing a lot, we'll have 4 under 4 by June in the next generation. So lots happening here. We would really like to know that, you know, the C word, the cure word, the fix the problem might be in our lifetime, which is pretty amazing. So thank you to everybody who has helped develop this technology and give us some hope for getting rid of HCM in another way rather than having to have your heart taken out. I don't necessarily recommend that. To those of you who are concerned about immunosuppression, next week it'll no, in 3 days, 4 days, it'll be 8 years. It's not that big of a thing. I bet you, if you know me, you know somebody who's been immunosuppressed for 8 years. You're talking about 60 days. You can do that standing on your head. It's easy. You're just not going to sleep a lot with the prednisone. That's about probably the worst thing you can say about it. So any questions, call the office. Interested in the trial, call the office, contact Tenaya directly. Look at clinicaltrials.gov, look at our trial finder. We are here to help make the process as easy as possible. Big thanks to my team, everybody who helped make everything happen tonight. Julie, who's sitting in the back end, making sure everybody's happy and got their technology working. Stacy, who helped organize. Olivia, who did the graphics. Thank you, thank you, thank you. Milind, Whit, any final words before we disconnect?
Thank you, Lisa, for hosting us, and thank you everyone for joining and participating. Thank you, obviously, Dr. Desai, and especially thank you to your patients for participating, and to the patients who have been eager to participate and have been unable for whatever reason.
Thank you again. Yeah, thanks, Whit, for co-hosting. So yeah, look, I have to say, you know, this is a team sport, and we have come a long way to go back. We are on our way, you know, we are on our way. We are going to science our way out of this disease. And the first steps have been taken. And, you know, again, it is not possible without the patient's trust in us. I mean, you know, look, at a personal level, you know, I think, yeah, it is pretty sobering that somebody would put their life and their trust in somebody like me. I mean, where I have come from, this is a pretty big deal. So I sincerely thank our patients, the system, the sponsors, and advocacy folks like you, Lisa, Wendy, for allowing us to be here today, to be able to do stuff so that we can talk about stuff like this.
I will say back in the day, patients would wait till the trial was done, till the data was published, until we got to communicate it to clinicians out in the field. Ladies and gentlemen, you're hearing late breaking. It doesn't get, okay, this all happened like right before Christmas, but none of you were listening to me around Christmas time. So we waited a little while. So you're paying attention now. So this doesn't happen in every space. Patients don't get this early data. So you're hearing it as it's breaking, as it's happening, so that you can make the decision for yourself and your family if it's right for you to do this trial. Or do you want to wait for some more data? Do you want to come in later? Or are you going to wait till it's approved and then look at it for yourself? All of those answers are right. We just want you to know what's available, how to look at the data, and ask any questions that you need to. This is a partnership. This is the ecosystem, and we all live here together. And hopefully, in another year or so, we'll have some other great data to bring you on this, and it's exciting times. I have said a lot since about 2022. It's a really good time to have a really crappy disease. There's so many things coming our way. So we need to make sure we still have the opportunity to do good science and bring good therapies to patients, because we've worked really, really hard for this for a long time. So pay attention to what's going on out there and use your voice when you need to. Thank you very much for joining us. And it was good to see so many familiar names and some new ones out there too. Hello, Harry. Everybody say hi, Harry. Hi, Yvonne. Hi, Wendy. Hi, everybody else. Those are the names that I just caught quickly. Hi, Rosemary, or one of our board members. So thank you all for joining us tonight. I feel like I'm on Romper Room, like looking in my magic looking glass. Thanks, guys.
All right. See you guys.
Bye-bye. We're going to stop on Facebook. And we're going to stop recording.